we've started studying for that exam. Let's see. We're also going to do the part 2 of the content check. So if I don't stop talking about 15 minutes till the end of class time, flag me down, stop me. It could be a problem though. Any questions about the exam before we get started? I know my last class had a lot of questions.
5 points.
45 points.
What's the weight of it?
The weight of it on your grade, 45 out of a total of like 300 total points, I think.
Is it curved?
It's not curved. Simply the amount of points you earn.
Is it like open-ended, multiple choice?
A little bit of everything. Yeah, so in fact on the study guide there's a link at the top of the study guide that brings you to basically the instructions that I would give you for each type of question you might see. So short answer, multiple choice, there might be some like interpret this figure, or circle this part of the figure. Right? Lots of different formats. Any other questions? All right. I know my other class had how many questions is it? I do not know because I have not yet made the exam. Probably less than 45 questions.
What would you put it as for like the level of difficulty?
Level of difficulty? Ooh. Good question, because it's so subjective. I would say comparison to your other exams, less difficult than the final, because the final is semi-cumulative. Third exam, maybe roughly the same as the third exam. Second exam, I think, is a little easier. So
far.
it's a lot of content. It's a lot of content. I don't know if I would say it's hard if you go through the study guide and you're prepared. You shouldn't have a problem with it, all right? But it is a lot of content. All right. I did have one question submitted to me that I wanted to answer before we got on to the rest of our lecture. I have the question, are the bonds between sugar molecules covalent or glycosidic? Well, it's both, right? So it's a covalent bond, but we specifically call it a glycosidic bond because it's between a sugar and another molecule. So yes, it's a covalent bond. Yes, it's a glycosidic bond. Right? So glycosidic bonds are covalent bonds. Not all covalent bonds are glycosidic because not all covalent bonds are between a sugar molecule and something else. Any questions here? All right, so we need to talk about energy and enzymes today. Last bit of material before your exam. Please let me know if something doesn't make sense as we're going through. So, energy. Energy is the ability to promote change or do work. Right? And there's two main types of energy. You've likely heard of these before. We have kinetic energy and potential energy. So kinetic energy is associated with movement. Right? So if we look at this archer up here, they have released the arrow. The arrow is going through the air towards the target. Right? That arrow is moving. That arrow has kinetic energy. Right? Versus potential energy. So potential energy is not associated with movement, but rather where that structure is or where the location is of an object.
This is my last question about the exam, but are we allowed to ask questions during the exam?
Clarifying questions, yes.
Okay.
Yep.
That was it.
All right, sounds good.
Thank you.
All right, so potential energy, if we look at this image up here, right, we're drawing back the arrow in the bow, right, because of the placement of the arrow, right, if you were to let go, it would have kinetic energy, but it's kind of like teetering on edge, right, it's about to have energy, but not quite moving yet, right, so that would be our potential energy. Now, in biology and chemistry, the potential energy we're often looking at is chemical energy, right? So this is a form of potential energy, energy due to the position and location, right? And specifically energy associated with bonds, right? So chemical energy is energy stored in bonds. When our two molecules are bonded together, that energy is there, right? It has the potential, but it's kind of stuck in that bond. Once that bond breaks, right, now that energy is released, and that energy can be used by the cell to perform a function. Right? So chemical energy, that energy stored in a bond, is a type of potential energy. There's a lot of other different types of energy, like light energy, heat energy. We talked about kinetic and chemical energy. Electrical energy has to deal with like ion gradients, so thinking about charges. Right? All these different forms of energy, they are important to biology, but we're going to really focus on this chemical energy for today. All right, in order to understand our chemical reactions and how energy works, we do have to go back to our laws of thermodynamics. So this is not a physics class. I am not a physicist, right? I'm going to keep this very simple. So first law of thermodynamics, you've likely heard it. Maybe you can repeat it, all right? It's a law of conservation of energy. Energy cannot be created or destroyed. It just can be transformed from one form or one type to another. Right? So we're never creating energy, we're never destroying energy, we're just changing the form in which it takes. Kind of a fun example of this, if we think about an ice cream cone, right? Lots of sugar in an ice cream cone. That sugar contains a lot of energy in their bonds, right? If we eat our ice cream cone, right, or maybe a toddler eats an ice cream cone, that energy is now converted, or that sugar is now converted into energy, right? And now that energy has become kinetic movement as a toddler is running around, or as you are riding a bicycle, right? We're just changing the form that this energy has taken on. Same if we think about plants and photosynthesis, right? They're harnessing energy from the sun, right? That energy from the sun is then going to be turned into sugars via photosynthesis. Right? And those sugars can store energy. It's a type of chemical energy because the sugars have energy in their bonds. So we're just changing the form. Now we have to go through one more law of thermodynamics, and that is the second law. Right? And this law states that the transfer of energy from one form to another increases the entropy. Right? So entropy is a word that describes the degree of disorder. So if I look at my image here, I have all these little purple particles. They're highly ordered into a sphere. If I increase the entropy, now all these particles, no longer in a sphere, they're chaotically scattered around. Right? There's more disorder to this system. We've increased the entropy. And typically as entropy increases, that's going to leave less available energy for other organisms to harness. Right? So we're kind of using up some of our available energy when we have more disorder occurring. A big pause here because I know in my last class too, there's a lot of writing that you're all doing for this slide. All right, so first law of thermodynamics, energy cannot be created or destroyed, we're just changing the form. Second law of thermodynamics is as energy transfers from one form to another, we're increasing the entropy, increasing the amount of disorder. All right, so just to put this into practice here, and this might not take the full 2 minutes, but I want you to answer the question, which has more entropy? The salt crystal at the bottom of a beaker or a water glass, or the water after the crystal has dissolved into its component parts of our two ions?
It's a 50-50 shot. Because it's started as one thing, bottom, and then it's
Yeah.
I did not get into the US. I just couldn't fall asleep last night. I kept waking up. Bro, my room has been like 90 degrees the whole time. It's really gotten hot in here. I wish my room was hot.
Alright, I'm guessing you've probably reached a decision by now. So, which one of these two has more entropy, the salt crystal or the dissolved in water? Yeah, the dissolved salt crystal in water. We're increasing the amount of chaos, the amount of disorder. So this one would have more entropy associated with it. Any questions? All right. So we're going to apply entropy now to thinking about chemical reactions and how they occur. So if we're thinking about a chemical reaction, and we're thinking about the amount of energy, right? So we're thinking about total energy. That's the product of the usable energy and unusable energy. Unusable energy being entropy, right? We've lost some of that energy to its disorder. The usable energy, on the other hand, is free energy. And free energy is often called the Gibbs free energy here, right, after the scientist that discovered this principle. And it's capital G when we see it shorthand. Right? So free energy is the amount of energy actually available to do the work. Right? Energy we can actually harness. So when we look at chemical reactions, we really want to look at the amount of free energy associated with these reactions. All right, so we're going to apply this concept of free energy to two different situations. Right, so a chemical reaction, it can either happen spontaneously, right? It can just go, or
Again.
no worries. So it can just go, or the reaction might need to cross a hurdle. It might need an input of something. Pretty sure this classroom and the one next door are like identical. So, yes. So an exothermic reaction is one that will happen spontaneously. It's just going to happen, right? We would say that the change in free energy, so this triangle here means change in. So change in free energy is less than 0, right? And that's key, right? Remember that number here, right? So if it's less than 0, the reaction is going to go. It's going to happen spontaneously. We would say it's exergic. And the resulting products are going to have less potential energy than the reactants. So if we look at an example up here, we have our reaction. We have our two reactants here, right? They're just going to simply follow along this reaction and turn into products. And in the meantime, it's going to release some energy. Now this energy might be released in the form of heat, right? Or in a chemical form, different means of energy here. But the big picture is that energy is being released. And we know this is going to happen spontaneously because the change in free energy is less than 0. Now that's opposite from a reaction that we would call endothermic. or endogonic. Right? In this reaction, it's not going to happen spontaneously. It's just not going to go, right? It needs an additional input of energy for this reaction to happen. So instead of energy being produced, now energy needs to be consumed before it can happen, right? And if we look at this delta G, we see it's now above 0. And again, that's key. So change in free energy is above 0. If we look at an example here, we have our reactants turning into products, but we need the input of energy to make this reaction happen. It's not going to happen on its own. If you think of like an analogy for these two types of reactions, think about your dorm room, right? Think about how little it takes to make a dorm room messy, right? You don't think about it, you just throw things down, it becomes cluttered, right? You make a mess with very little energy associated with that, right? You don't have to do much for your dorm room to be messy. But in order to clean up your dorm room, right, now you have to put effort into it, right? You have to work to clean up your dorm room. So that would be similar to this endogonic reaction, right? You need to put energy into it in order to make it happen, to make that dorm clean again. All right, and this is where we need to start talking about ATP. So we've mentioned ATP multiple times, and I mentioned how it provides energy to a reaction, right? Well, this is the reasoning why. So if we look at the molecule of ATP, it has 3 phosphate groups, right? And the bonds holding these phosphate groups together are full of potential energy. So if we were to break this bond through the process of hydrolysis, so we're adding a water molecule to the equation, it's breaking apart the bond, that releases the energy, right? And that released energy can then be used to power a cellular process. Think about active transport, right? We need ATP in order to move solutes against its gradient, right? The energy is physically coming from the breaking of a bond in one of these phosphate groups. Now, if we look at the change in free energy, we see that it's negative 7.3. So this means it's going to just happen. It's going to happen spontaneously. So we don't need to add more energy to the system in order for ATP to break apart. All right, so because this breaking apart of ATP has this negative delta G, it could be used to be coupled with other reactions to make them happen. So let's look at a reaction. We're going to look at glucose and phosphate. We're going to combine them together to make glucose 6-phosphate plus a molecule of water. That reaction alone is not going to happen spontaneously, because if we look at that change in free energy, it's above 0, right? It's 3.3. That means it needs energy to work. It's not just going to happen. But we know ATP, if we break ATP apart, right, that reaction in itself is a negative 7.3. It happens spontaneously. So in order to make this first reaction go, we can simply combine those two reactions together. We can couple them together. And if we add the change in free energy from both reactions, right, we get a negative value of -4, right? And if it's a negative value, we know it's spontaneous. So we're combining a reaction that's not going to happen spontaneous with a reaction that does happen spontaneous, right? And if we add our change in free energy together, the overall number is a negative, so it's going to just happen, right? ATP is coupled in a lot of reactions in this manner. All right, so if we're thinking about reactions, I just want to kind of put it out there that it's not just what the change of free energy is going to determine the rate, right? There's other factors there determining how fast this reaction might occur. One of those factors is temperature, right? So typically in a solution, molecules will be moving around, right? If we increase the temperature, molecules are going to move faster. They're going to collide more often, making it possible for reactions to occur more frequently. Same thing if we add in more of the solutes, right? If we add in and increase the concentration of reactants, there's just more molecules, more possibilities for collisions to occur. So these reactions, again, they're going to happen faster. And we can see that in this graph here. So we have mass of a product over an amount of time here. The concentration is very concentrated, right? A lot of solutes. That reaction rate is going to be much quicker than if there were fewer of those molecules in our solution. All right, let's review what we've learned. So, 2 questions here. I want you to look at these 2 images. Which one is going to happen spontaneously? And then I want you to think about this reaction, so the decomposition of calcium carbonate. Looking at its change of free energy, which reaction can it be coupled with to make it spontaneous?
I need to give my brain a second.
Go on here. Relax, quiz. Take care of.
Yeah.
I'm assuming you probably haven't had this last Okay. All right. I was the same way. I wrote so much code.
Tonic. Answer to the
What's negative? It just means that it doesn't have to process. It has enough energy in and of itself. On the one versus if you were these are sliding down the hill. You're sliding down the hill, but there was like a fall. I don't like that. Remove that roll before you use energy to go. Yes. Yeah. All right, let's do by show of fingers here. this graph, is it going to be 1 or the second one, so 1 or 2, that will happen spontaneously? It would be 2, yes, because if we look at the change of free energy, it's less than 0. What about this next question? Which of these 4 reactions could we couple with the decomposition of calcium carbonate to make it spontaneous? A. Yeah, A is the only one that if you add the values together, we would still have a negative number. So a lot of concepts here, but it's all leading up to kind of this paying attention to the free energy. Is it less than or greater than 0? Any questions before we move on? All right, well, let's see how enzymes play a role here. So, a reaction can just happen, and it can be spontaneous, right? But if it's spontaneous, it's not necessarily a fast reaction. In order for a reaction to speed up even faster, cells will use enzymes. So enzymes are catalysts, right? So it's an agent that helps speed up a chemical reaction, and it's not being used up in the process. Right? So we can use the same enzyme over and over and over and over. An enzyme refers to a catalyst that's a protein, right? So made up of amino acids. Right? So a protein catalyst is what our cells use to help speed up and increase the rate of our chemical reactions. So if we look at our diagram here, we have our enzyme. Our substrate is coming into contact with our enzyme. Notice how the groove in the enzyme is very specific to the groove of the substrate there. They match together perfectly. The reaction occurs, and then the products are released. So enzymes are a great example of structure equaling function. Okay? Because they have a structure that is very, very specific. So if we look at enzyme, right, we have our blue enzyme here. It's going to have an active site. And this active site is typically going to look like a groove. Okay? And it's very specific to a certain type of molecule. That molecule we would call a substrate. One word. Oh, I went the wrong way. So structure equaling function has a specific groove. So if we look at this example here, our specific groove here, this would be our active site. So our active site is where specifically that substrate binds. The substrate is the molecule that will undergo the chemical reaction. Now, when they're bonded together, right, so when the substrate is inside that active site, we would call it the enzyme-substrate complex. So this active site is very specific. Each enzyme will have a different shape, and thus a different shaped active site. As the pen's curiously moving, so I'm just going to take a quick pause. All right, so enzymes with their active site, they are highly specialized, right? So structure equals function. The structure of each individual enzyme is specific to a particular substrate, right? So a substrate acts upon a very specific molecule. It's designed specifically for one type of substrate. If we have an enzyme trying to react with a different substrate, it's not going to fit, right? It's not molded for that substrate, and that reaction is not going to work very well, right? And so this specificity really allows the cell to control what is happening on the inside. So say it wants to increase the reaction rate of one reaction, but not all the reactions in the cell. It can do that because an enzyme is specific to one reaction, right? So by using one enzyme for one reaction, it's just increasing that one reaction instead of increasing every single reaction within the cell, because it's so specific to the molecule that it's acting upon. All right, I have a nice video here that gives some great analogies to why enzymes actually speed up reactions.
Right now, trillions of chemical reactions are humming away in the cells of your body. You never feel them, but without these reactions, you wouldn't be alive. Unfortunately, each of those reactions needs some help. You see, most molecules are stable. They're happy just the way they are. The atoms in them are all bound up and friendly with one another and would prefer to stay that way. Problem is, for a chemical reaction to happen, the atoms that make up those stable molecules need to break away from their friends and go buddy up with another atom. This breakup is where the molecules need a hand. This initial kickstart is known as activation energy. It's used to destabilize the molecule, to push the bonds between the atoms to a place where they are ready to break. This unstable state is known as the molecule's transition state. Once the transition state has been achieved, the atoms become willing to leave their current molecular friends and go make new friends elsewhere. Once they're convinced, it's a piece of cake. Bonds break, atoms rearrange, and the rest of the reaction happens automatically. After that first push, the body doesn't need to put in any more energy to help the reaction along. Left alone, most of these reactions would be very slow, because it takes quite a while to build up the activation energy the molecules need to get started. Enter the enzyme. Enzymes are proteins that speed up or catalyze reactions by lowering the activation energy. They make it easier for the molecule, also known as a substrate, to get to the transition state. You can think of a reaction like a race. Some racers are running alone, while others have teammates to help them. Meet Sam, the substrate. His team is the Mod Squad. Together, his team is able to get to the finish faster, using less energy. There are 4 special enzymes in Sam's team. Each has a different strategy for lowering the energy it takes to get going and speeding up the pace to get the mods to the finish line. The M stands for microenvironment. This enzyme creates a tiny, special environment for the substrate, resulting in a faster reaction time. He runs ahead of the pack, flattening out bumps in the road and misting cool water on his team of molecules. O is for orientation. Sometimes 2 molecules must be positioned just right before they will react. Like a friend at the finish line, the O enzyme provides its molecules with specially shaped spaces that allow the substrates to interact in just the right way. D stands for direct participation. Every now and again, a little muscle is needed, and when his teammates are struggling to finish the race, teammate D is there to pick them up and carry them over the line. Finally, S is for straining bonds. This guy pushes the team through some serious flexibility exercises. Splits, lunges, back bends, the works. Sometimes his substrate teammates just need to be stressed and flexed into their transition state. So that's it. Remember that all reactions need energy to get going. This energy is known as the activation energy. Enzymes lower that activation energy and speed the reaction through team mods: microenvironment, orientation, direct participation, and straining bonds.
All right, so a reaction can be spontaneously and just happen, but it still needs to overcome a little bit of a hurdle, and that hurdle is the activation energy. Now, for spontaneous reactions, they can overcome that hurdle on their own, but it's very slow. And this is where enzymes play a role, right? These enzymes lower this activation energy, the energy needed to get these bonds or get these molecules close enough together, right, that they can interact. Right? And if we think about it, if we have 2 molecules that are going to react with each other, right, what is surrounding every atom of that molecule? It's a negatively charged electron. And so if we have negatively charged electrons getting close to other negatively charged electrons, right, they're like charges, they want to repel each other, right? So it has to have a little bit of a push for those molecules to get close enough for them to actually react, right? And this is that transition state. This is where it kind of stresses out those molecules to the point where they can rearrange their bonds to form their products. Right? Common ways to overcome this activation energy, well, you could add heat, right? But enzymes are really the key players here. Right? They're lowering that amount of energy needed to stress out our molecules so that they can rearrange into their products. We saw in the video some common ways that enzyme lowers this activation energy. One way is just by stressing those bonds, by straining the bonds, right? So think about you're a coach and you're trying to get your players into shape, right? You have to do some activities that might not be fun at the time, stresses you out a little bit, but will help you in the long run. Right? So physical strain, we're putting physical stress on the substrates in order to get those bonds to break. We can also position these two molecules close enough to each other in the proper orientation that bonding can actually occur. If we think about the analogy they made of the race, right? That teammate at the end was laying down, now we want him to stand up, right? So the enzyme can help it stand up so it's in the proper orientation for them to actually interact. And we can also change the local environment, right? So just like that teammate was running ahead and filling all the holes on the road there, right? Making that environment pleasant, right? That's essentially what's happening here. Inside of our active site, this is a very localized environment. That environment is being catered towards that substrate to make it just right for those bonds to break and rearrange. Right, so that would be changing our local environment. That's different from changing the environment that the enzyme is actually in. So the local environment just refers to this active site where the substrates are binding with the enzyme. But the enzymes themselves will have a range of environments that they work well in. And that really depends on what organism they're found in. So if we look at a couple of examples here, right, we have bacteria that lives in a cool and a neutral environment, and bacteria that lives in a hot and acidic environment. Right? If we look at the relative enzyme activity, and this enzyme is chitinase here, if we look at the relative activity, we see that for a bacteria that's used to a cool environment, that activity is going to be highest at a lower temperature than the enzyme that's used to living in a hot environment. Same with the pH, right? If an enzyme is living in a bacteria that lives in this acidic environment, it's going to work better at more acidic of a pH than this bacteria from a cool and neutral environment. Right, so not only do they create specialized local environments in their active site, but the environment surrounding them can influence how well an enzyme works. And if we think about proteins, we know they can denature, right? And if we denature a protein, there it loses its shape. Shape equals function. These enzymes can no longer work. All right, so let's think about a series of reactions here. We have this pathway at the bottom, right? Multiple substrates, multiple enzymes. I want to know what would happen if this enzyme, enzyme C, all of a sudden becomes non-functional. Right? Maybe it's been denatured in some way. How would that affect the rate? And how would that affect substrates or production of products? Cool.
Well, you still my brain is taking it too long to process this. So yeah, it wouldn't be able to get the product because there's not an enzyme. There's not the enzyme C. So now just go 1A, 2B, 3, 4.
It'd be Sept.
Yeah. Which doesn't, yeah. There would be too much substrate. It wouldn't turn out the way maybe, I don't know, that makes sense, but it kind of doesn't.
Yes, question.
It makes
So you
sense.
know, enzyme C is disrupted.
I would say that's maybe a different question altogether.
Okay.
So right now we're just saying enzyme C. If it's not working, likely it is changing the structure. It's likely becoming nature.
So it slows down the rate. That was a great.
How are you feeling?
There's a lot going on. I have
This
a
exam and other things.
I have this exam, and then I have a psychology exam, and then I have a rough draft
Woohoo! Lots to do.
I may or may not.
I wanted to do it on Friday, but I don't know if you're ready.
So you have them just for fun.
All right, so let's think about this chain of reactions here. If enzyme C is no longer working, what is happening to the rate of our reaction?
Slowing down.
It's slowing down. Is it stopping?
No.
No.
No, because it could still happen without enzymes, just much more slowly. What might happen to the accumulation of substrates? Would they start to accumulate, or would these also slow down? They would accumulate. Yeah, enzymes A and B, they're working, right? They're still creating their substrates, right? Essentially, it would accumulate until it hits this bottleneck, which is enzyme C, and things go much more slowly. What about the production of products? Is that going to increase, decrease, stay the same? Decrease, yeah, because we're slowing down the rate of the reaction. It is also going to decrease the rate at which these products are being made. Any questions about enzymes? All right. I have 3 more slides here, but I made the decision in my last class that these slides I am not going to test you on, and so I'm just going to kind of skip over them. I inputted the answers onto the versions of the slides that you have, so if you downloaded it earlier, you might not see it pop up. You might have to go back in. Right? It's really just talking about enzyme reactions and different inhibitors that could lower the rates of reactions. We're skipping over this. We're not going to go into this. You can look at it at your own time. So I want to move on to the content check. Before we move on, I do want to say that the homework for Wednesday, it's that scientific literacy assignment. On Wednesday, we'll have a short discussion about it, and then the rest of the time will be used to study for the exam. I'm going to give you like a practice mini exam to do, and